Can be recorded and posted in YouTube this time. So it's State Micro, which is ST capital ATE Micro, I think, or yeah, I think that's what I did. So you can definitely search it. I think I sent the link, didn't I, to the channel? Share screen. Go move the camera, Dr. Wall. Move the camera to the board.
It was adjusting on its own. It's like whenever she would come to the board, it would adjust.
Hello. I'm going the wrong way. She did. It was all fixed and zoomed in, and she's like, "I don't like it zoomed." Yeah. And let's see if I can zoom. Oh yeah. Look at that. Oops. Okay. Let's get on with it, right? So again, this should, just double check, this should correspond to 590 in the text. Pictures. Go. 590 is still kind of the introduction of everything. Really want to move over. There, intestinal protozoa, that's right. Chapter 47. So that's a big page gap there, you see. Okay, all right, y'all ready? So it's Friday. Are y'all gonna like Friday being lectured? We had Friday immunology, right? So nothing really new. Are y'all gonna like lecture without lab? Is that gonna work? Does it feel like you're not just like, ooh, we went from one to the other? You feel like you got some space? Because I'm kind of feeling that already. Just week one, I'm feeling like, oh, don't feel so rushed to get into lecture before a lab. It seems to have a little time. So we're going to start with the pathogenic amoebas. So that's our what we're going to do. If you look at your outline, it should talk about intestinal protozoa, and amoebas are part of this. Flagellates are part of this, and ciliates are part of this. So, but we're going to start with the amoebas. So we have a really good video to watch. Not the parasite eaten alive videos, but actually a news story about a pathogenic amoeba that we're going to cover today. So we kind of made national news a few years ago. So intestinal protozoa, protozoans are parasites distributed worldwide. The sanitation, well, I think we've covered that enough that you know that if you have poor sanitation, there's chances of spreading, coming in contact, infecting water supply, getting ingested. So hopefully you got a really good idea of what ingesting a cyst can do from yesterday's lab. So temperate climates, the United States, more common in rural areas under crowded conditions and most often among the poor. So I don't think anybody could look at this state and say, oh, that doesn't describe us, right? So these are your patients, the patients that could come to your hospital tomorrow, could show up and maybe have an intestinal protozoa. So the thing about protozoa is they have two stages in life, and this is where we introduce these first. Common theme that we're going to have all throughout parasitology class is the trophozoite. Okay, that is kind of like the adult form kind of like the mobile form. So these are moving, actively feeding. So these are live parasite organisms, and they actually can divide at the trophozoite stage. But on the good side, if there is a good side, they're very fragile. So just if you'll read through some of the things we're going to talk about today, just putting them in saline can kill them. Okay, just introducing a different environment to them can kill them and actually get rid of them. So they don't survive outside the body. If they do, it's only for a very short time. So the trophozoite is that adult form kind of thing after the cyst has gotten in and has developed into this adult. The cysts, on the other hand, are kind of like the eggs. Okay, so they are the stage which are more resistant to the environment, so it's very hard to get rid of or kill them. They're going to be, just like you saw yesterday, if they get encysted into a meat, they get into a water supply, that's how they're transferred to the next host. This stage allows survival outside the body. So if it is like you saw from our cow example yesterday, human fertilization, maybe not the best thing to do to your garden outside, especially if you got animals that can get out there. Like I think about we had a cow yesterday, but something like a dog. I mean, how many dogs go out and eat grass? Mine does. About every other day, she goes out and starts, munching on grass that could have feces from another animal on it, maybe has a cyst in it. She ingests that, and then all of a sudden she's inside the house, she's on the couch, she's in a bed. Just something to think about. So the cyst is known as the infective stage. So that's very key to remember and put in your memory for test. Protozoan parasites in the cyst form, which is acquired most often by ingestion. We think we established contaminated food and water. Unlikely the troph form would survive the acid environment of the stomach. So a troph, if a troph has infected your water, you ingest that troph, feces, contaminated water, probably not going to make it through that part. You're not going to start getting the troph and then all of a sudden, even to the point where we see solid stools have trophs, okay? So the troph is not the cyst form. So this is what we're going to look at a lot. We're going to have CDC examples of life cycles. So these are very simple, but they contain a lot of good information. And one of the things that we see here is that this would be our cyst form, and this is our troph. Okay, very, one looks like it's round, nicely compact. The other looks like it could be moving around. An amoeba can move, so kind of has that pseudopod look that, it's going to be moving and invading the tissue of the intestine. So this, what you would see is the feces comes out, right? Step one, there's the feces containing cyst and troph, okay? And the cyst and troph are passed, okay? But not only the cyst is going to be transferred into the next host. So once you see that, then you see all these different places that and this is, I want to say, it's the entamoeba life cycle here. And what you'll see is the infective stage, okay? Infective stage is the cyst, okay? So it gets the eye with the triangle. Diagnostic stage, we're going to be doing this with feces. We're going to be looking for both cyst and troph in feces. So that's how we would diagnostic with a direct wet mount. So you may see something like non-invasive colonization, intestinal disease. That would be our bees invading the intestine, the large intestine here, causing bloody diarrhea as it invades the wall of the colon. Extraintestinal disease, meaning it's spreading to the liver, spreading to the lungs, spreading to the brain. You can see that they can be found in those different areas. So this kind of gives you an idea, I hope, to see a simple way. And then at the bottom, you see the cyst. You see this diagnostic stage of the different forms of the cyst. So you'll see that it may say it's 1 to 4 nuclei. And that's going to show right here in the cyst form. This is a cyst with 1 nuclei. This is a cyst with 2. That's a cyst with 4, and a cyst with 4. The troph, on the other hand, is probably only going to have 1, may have 2, but usually you're going to see it with 1. And then you hopefully see the different shape to help you diagnose differences in a troph and a cyst. So again, when we saw those white blood cell inclusions yesterday in lab for pseudoparasites, when you see these maybe side by side, you get to see that. Maybe a confusion. The diagnostic stage, again, troph or cyst, either one, both, that are recovered from the patient specimen. And either form should be identified and reported because parasites colonize certain areas of the body. Oh, my camera just did a funny there. You'll notice that. Is it sensing me, you think? Is it?
Yeah.
Oh, wow. I didn't know that. I didn't know it could do that.
It was at the point it zoomed in on you.
Oh, it moved when I went to the board? Wow. That's I didn't know it could do that. Oh, okay. Something that zooms like, what are you talking about? There's maybe seeing it over to the side. The location within the host depends upon the species of parasite. So different parasites will go to different areas. Just put it that way. So transmission of intestinal parasites associated, again, poor sanitation, overcrowding. You know, I once worked at a hospital where we were the prison hospital. Anything that went wrong at Cummins Prison came to our hospital ER. So definitely we could see prison patients come to you, depending on where you're working, what hospital's covering that. Sewage contamination of drinking water. Since most parasites are GI parasites, meaning gastrointestinal, the specimen of choice, which I think you've got that. I hope by week 1, y'all have got this down, that it is the stool specimen when it comes to parasitology. Careful examination of the stool will often allow diagnosis to be made. And don't sell yourself short, right? When you go out to clinical and they're like, oh, we got a diarrhea sample, all they want to do is a C. diff, and you go, you know, let me try my direct wet mount on this. Go ahead, take a drop of stool, put it on a slide, go review it. That's practice. Even though it may not be a parasitic infection, but who knows? At least you get some practice in calling them negative. And then who knows, you may just be the eyes that you're fresh out of class and you go in there and you're like, that looks like a trophozoite. That looks like a cyst. That doesn't look like regular stuff out of stool. And because you're going to be probably as I said, the freshest eyes when it comes to clinical in your hospital as far as looking at stool. So how do we prepare? We're going to do direct wet mounts. We'll be doing that on Tuesday. We're going to evaluate to see if there's any motility. Our samples we're just going to leave that door open, that book. You're just going to leave that open, what we're going to see on a direct wet mounts, because I have not gone through all those samples. So we're going to see them together on Tuesday. We'll use our first one and see what our saline direct wet mount without iodine, okay? We'll do that, and then we'll do one with iodine. Concentration procedures, this is if we just, hey, it's such a small number, maybe we want to concentrate it, bring it down, spin it down to recover the cyst, particularly useful when it occurs in low numbers. And then we can do permanent stain, you know, with the iodine or with the trichrome to save it. Like, oh yeah, we're going to put that over here. That is a positive parasite found or positive cyst found in the stool. And then here, serology, it is used. We're going to see it today in lecture with one of them today, but rarely used to diagnose infections of intestinal parasites. But there are serological tests that you can look and use for antigen testing for certain ones. And then what's toxoplasmosis?
Isn't that the thing pregnant ladies
Toxoplasma gondii? Are you over there? Okay. Toxoplasmosis says an exception. So that would be one that would have the serological test, right? What is toxoplasmosis? I think you're on the right step. Somebody, Allie, you looking it up for me?
Yeah.
Okay. Is it gondii?
Parasite found in cat feces. and
There you go.
contaminated food.
Yay. You all knew that, right? Not just Allison. Okay. Back to trophozoites. The type of motility may be characteristic of certain species. The number of nuclei is another distinction. So we're either going to be mononucleated or binucleated with the trophs, where the cyst is usually going to could have one or more than two. And we covered that. Now, then we're going to have parts. And parts is this idea that we have the presence of peripheral chromatin. And what we want to get, we can go back just a little bit, bring this back. So if we look at this trophozoite here, okay, so this is the nucleus. Okay, and we're going to see this a lot. We're going to see that we have a center dot, okay? We have an outer membrane, okay, the nuclear membrane. And then around that membrane, we can have this chromatin, peripheral chromatin up against that nuclear membrane, adding thickness to it, look to it. Okay, so this is all the genetics. This is all DNA, RNA, up against that chromatin, chromosomal material in the parasite itself. And then the karyosome is in that center dot, okay? And that's just a clump of chromatin in the middle. So we're gonna see that with these definitions. Let's go back, sorry. So the cytoplasm, well, did we start? Did we miss? Yeah, here it is. The presence of peripheral chromatin depends on the species. So you're gonna hear a description. It might say, oh, there's peripheral chromatin in the nucleus. The pattern of peripheral chromatin is uniformly or uneven. The appearance of the karyosome, which is that small, compact, rounded in the middle, right, in the middle of the nucleus, it's still chromatin material, it's still DNA in that. So that's the nucleus. The cytoplasm, right, inside the trophozoite or inside the cyst, but outside of the nucleus, may appear smooth, it may appear coarse, maybe hyaline. Okay, you may also look for the presence of inclusions inside these trophozoites. So a trophozoite might have bacteria because they're feeding, okay? They're feeding. It might have yeast, organic debris, or even better for us, it may have a red blood cell. So what I want you to do, we had this wild day in lab last year that we learned quite a bit from. So what I want you to do is think about your smears that you looked at in hematology. Just start thinking about that right now. And I know you had this on a test question, so tell me, somebody, the size of a red blood cell.
So it's like the size of a resting lymph, but like the exact
I need
numbers.
numbers.
No.
In parasitology, I need a number.
Isn't it like 7 to 12?
7, how about 6 to 8? Does that sound better?
What is it, pico?
Micrometers, okay? So what I gotta do is I gotta give you something that you've seen over and over and over, which is what? Red blood cell. Okay, so we're about to see the size of these parasites, whether it be the cysts or the trophs. So you can imagine if I can engulf a red blood cell, 1 or 2, then I'm probably bigger than a red blood cell, right? So when we think about the size of the parasite, when we see these numbers, I want you just to go back and just simply, because we had this whole lab where we were measuring with the microscope, micron, 1, 2, 3, 4s, and oh my gosh, it just got a mess and we just fell apart and the train crashed. Okay, it came off the track and we crashed that day in lab. Not gonna do that again. Okay, so you'll see that in the lab folder for exercise. We're just going to start off saying we're going to compare everything to a red blood cell. Because you've seen that. You're going to see that over and over and over, whether it's at 10x, 40x, all immersion, right? You've seen the red blood cell. So compare everything that you think about, everything that we're going to size up in parasitology this semester. I want you to compare it just simply to the red blood cell. So 6 to 8 micrometer red blood cell. Keep that in mind as we throw these other numbers up here. Okay. The cyst stage, remember this is the infectious stage where you get the next host. The number of nuclei vary, each species. So we have character of nucleus in most cases, somewhat similar. Chromatin bodies, okay, we're going to see these chromatin bodies. These are going to be in the cyst, okay, outside the nucleus, and they're going to be darkly staining and big, okay. They're still going to be inside the cyst, but these are crystalline RNA, and they're found in immature cyst. There may be something known as a glycogen vacuole in the cyst, and hopefully you can start to see some of the egg thinking, right, with a glycogen vacuole. Hey, I need some energy as I'm a cyst. The shape and size varies, and we'll see the diagnostic stages as we move through. Okay, so that's the intro to kind of some of the definitions, kind of the things to come. We're just going to, every time we talk about a new parasite, we're going to hit on those same characteristics. We're going to talk about the cyst. We're going to talk about the troph if it has those forms. Okay, and we're going to see how they change and look, so that when you saw Entamoeba histolytica yesterday on that last slide that I threw up there, number 38, whatever it was. So think of that today, because that's where we're headed. So the first one, Entamoeba histolytica. I'm going to start with the troph, or is that trophozoite, sorry. So the trophozoite, the nucleus, the nuclear membrane appears to be thin. The peripheral chromatin is uniform, small in size, and evenly distributed. So we're not going to see this chromatin moving away from the nuclear membrane, but into the nucleus itself. It's going to be even. The karyosome is the small, round, compact, centrally located dot. Okay, so we're going to have this nucleus, and we're going to have the karyosome right in the middle, and then the chromatin's on the outside of the nuclear membrane. And that's going to be in the troph. Is everybody good with that? Is everybody starting to pick up these labels and names? So if you saw one, you can label it. All right. In the cytoplasm, fine granular appearance, hyaline clear ectoplasm. The inclusion with the histolytica is it ingests red blood cells, and that's going to distinguish it from the next Entamoeba we're going to look at. It does not. The next one does not ingest red blood cells, so this needs to be double lined, double circled, whatever you want to say. When you get the test question that says, how do you distinguish histolytica from another Acanthamoeba? Okay, you're going to say, it's because it ingests red blood cells. Look at the size of the troph. Average size is 20 micrometers. Double a red blood cell, right? Size range could be anywhere from 12, still bigger than a red blood cell, up to 60, okay? 6 times a red blood cell if you want to look at it. Motility, they can move, directional with long finger-like pseudopodia, false feet, all right? So the cyst, so that's the troph, remember that's the non-infectious stage, that's the invader. Once it's in, that's the one causing the damage to the intestine. The cyst, on the other hand, usually round, recognized by their presence of hyaline cyst wall. They have nucleus, they may have 1 to 4. We pointed that out earlier. The peripheral chromatin is similar to that of the troph, so it's kind of uniformly, it's not really, boom, moving away from the outer membrane. The karyosome is centrally located, And then the cytoplasm around the nucleus, inside the wall of the cyst, is finely granular. Inclusion. Trophozoites extrude all ingested material when they encyst. So when the trophozoite moves to the cyst, okay, it spits out the red blood cells that it ingested while it was eating. So you won't find red blood cells in the cyst form. You will find chromatin bodies though. Chromatin bodies are long bars of rounded ends, look like cigars, stained bright red with trichrome or darker with iodine. They're diagnostic for the whole group of entamoeba cysts. The histolyticas, the Hartmannis, and the dispar. The glycogen vacuole, again, that's only in the cyst, found in immature cyst. Pushes the nucleus to one side, so it kind of fills up the cytoplasm with this big glycogen vacuole for storage, energy. And then the cysts range 10 to 20. So not much different, right? So approximate size of the troph, 20. Approximate size of the cyst, maybe a little smaller, 10 to 20. So here are the nuclei. This is what we're talking about with the variations in the nuclei. So what is all together? Follow. What is this? What is this bullseye in the middle? We were labeling it. Y'all shook your heads. You said you could label this. What is that in the center? Karyosome. Correct. What is this on the outer edge of the nuclear membrane? What would you label that as?
Cytoplasm.
Finely distributed on the outer edge. See, it's different at all different stages here. What would you label that as? This is the karyosome. This is the nucleus. What is this?
The peripheral chromatin.
Peripheral chromatin, correct. Okay. So we got that down. If you hadn't got it, if you're like looking at me confused and you're like, just go home and label one. That's not hard, just so we can understand where we are with the description. Okay. So if this was a cyst, we could have either 1 or 4. And we're going to see pictures of that. So this puts up the trophs and the cyst of histolytica together. Okay, so the easiest thing to do is, let's start here with number 5. So this is a cyst, right, round. That's that that's been extruded to this periphery. What is that over here? Y'all see me on, maybe not good, but what's this right here again? The whole thing. The nucleus, that's the nucleus. Peripheral chromatin, karyosome in the middle. So this is what? What is this big blob of stored energy right here? The glycogen vacuole. Good. Okay. So if we went to just the normal cyst over here, you would see 1 nucleus, peripheral chromatin, karyosome in the middle. What would this cigar-shaped object out here be? Chromatin body, right? So here's a cyst with 4 nuclei. That's not a troph. It's got 4. Troph won't have 1 or 2. Here is what? What's that? Number 1. Troph. Yes. Okay. Number 2. What's number 2? Is that round? This one here. Is it round? Got 1 nucleus. It's not round. Troph. Number 3. Is that round? troph. Okay, 4. Say 4. Containing. Remember what I said we were going to measure our size of our stuff with? What is this in here, you think? RBC, so that's a troph. The cyst will not have red blood cells. Okay. This is a great one to work through just to get the comparison on what we're looking at. So when you look at a slide, if it says, hey, this Entamoeba histolytica sample has trophs and cysts in it, hopefully you're going to see the difference. That's what we're waiting on Tuesday to see if we can do this. So here are some of the slides from our slideshow. Anybody want to take a shot? This is a troph or a cyst? Troph. 1 nucleus. Got about what? How many red blood cells? 6. All right. Yay. One better. Trichrome stain. Troph or cyst? Troph. That baby's moving. All right. Got 1 nucleus. Got about how many red blood cells? 4 or 5. One of them looks like it's struggling up there at the top. It might be the second nucleus, right? Doesn't kind of got big clump in there. I wouldn't say that's my karyosome, right? So, but it, you know, if you had to really hammer home, cyst or troph? Cyst, round, right? Cyst or troph? Cyst, probably a glycogen vacuole maybe here. What's this out here? Chromatin body. Cyst or troph? Cyst, why? Chromatin bodies and 3 nucleus that I can see. Cyst or troph? Cyst, 4 nucleus. Round. What do you think we got here? Got a side-by-side comparison. Pretty much the same size, right? So I don't think that's a red blood cell inclusion, but you got to make sure it's chromatin look to it. Kind of roundish. This one's kind of moving out, elongating. 1 big nucleus there with the karyosome in the middle. This peripheral chromatin out here. You are already experts, aren't you? Can't wait. All right. Good job. All right, clinical significance of histolytica, 85 to 90% are asymptomatic. So you're infected, the intestine, not showing any signs or symptoms. Most common disease is amoebic dysentery. Dysentery is diarrhea, watery diarrhea, pain, cramping, flatulence. Everybody know what flatulence is? It's kind of like a science word for gas. Yeah, gas. Specific body area, the colon. The amoebic dysentery is associated with invasion of the bowel wall. So once we start invading the bowel wall, that's when we start getting the blood and water into the stool. So how would we demonstrate it? Direct wet mouth. Look for the progressive motility. So if we got trophs, they're going to be going. We're moving. Okay. We got a good wet mouth from a stool. Concentration, look for typical quadrinucleated cyst. Trophs may be lice because they're just too fragile. And then the trichrome stain, we look for both troph and cyst. So that's our diagnostic stage. That was that very first thing we saw, the diagnostic stage from the CDC. Life cycle is that we use both troph and cyst for diagnosis. These aren't too bad. This is just a colon wall showing you blood, invasion, infection, irritation. Another invasion of the intestinal wall. So it's cross section. So that's where it wants to be, but it can move, right? It can be in the liver. So if it spreads through the intestine to the liver through the blood, that's how it gets transferred up to the liver. Tenderness and enlargement of the liver. You may see abscesses in the liver on x-ray. A biopsy of the liver may show trophs. And, you know, if we're going to cannot reliably recover trophs from this aspiration. So if we saw an abscess on the liver and your doctor goes, well, we're just going to go in there and we're going to drain that abscess and see what's in there. They call that gump that they find in the liver anchovy paste. So it has this brownish, light brownish, gooey look to it. And we may not be able to recover the trophs from that. So we're going to demonstrate this. A saline wet mount will take away the trophs but leave the cyst. And then we can do an H&E stain for the necrotic margin of the abscess. So this is, we're not going to be doing this thing. This would be more of a pathology. Look to it. You may be taking that paste and spreading it on a slide for the pathologist. I don't know. Y'all do any H&Es of liver abscesses?
I know we do H&E stains, but I don't do that. Our histotechs do that, but I don't know now. We always do that.
Have you seen the H&E stain done? Okay. Again, this would be our liver abscess. What it looks like to me. All right. So again, what do you think this is? Troph or cyst? Troph. It can actually go to the brain due to the spread. Anything that gets in the bloodstream can get taken to the brain. Venereal disease can be passed along through sexual contact, whether it be heterosexual or homosexual. I'm not sure if that's a leg or a side or an abscess of some kind. All right, so again, diagnostic stage, we find cyst or trophs. The infective stage is the cyst. Yay. Acute disease, there's little chance of transmission due to passage of trophs, forms only loose watery stools. The chronic disease has troph and cyst at different times. So usually an acute person undergoing an acute stage just has the troph forms invading the intestine and will not have the cyst to transfer. If you have a chronic infection of histolytica, that's where the cyst will have time to form and then get transferred. All right, diaspar, Entamoeba diaspar. It is the non-pathogenic. So Entamoeba histolytica is pathogenic. Diaspar is non-pathogenic. It's indistinguishable from histolytica. So if you had the trophs, yeah, you can't tell any difference, right? But there is one difference if you do have a chance, and it's just it's not the definitive way you would do it, but if you had a histolytica and you saw red blood cell inclusions, you'd go, yeah, that's histolytica troph. But I don't know if you had diaspar, would you know, all those trophs would not have any red blood cells. They don't have any inclusions. So that's one thing to remember with Entamoeba diaspar. Size, 12 to 40, still bigger than a red blood cell. Motility, directional, disease does not cause disease or invade the tissue. They're morphologically indistinguishable, histolytica and diaspar. And you can't do it microscopically alone, but there is reasons unless you saw the red blood cells inclusions. So on a test, right, on the midterm slideshow, if I put up this and I said, okay, is this Entamoeba histolytica or diaspar, what would you say? It would have to be marked as can't tell, right? Got to go with both until we see what? Red blood cell inclusions. Again, can't distinguish that one from the two. So those would probably go together. And that brings us to this. Naegleria fowleri. Okay, this is the brain-eating amoeba, if you've ever heard of it as that. So this one, ignored all the way till summertime. And we have had cases in Arkansas in the recent years. One of my longtime childhood memories of going out to a water park south of Little Rock. That's where our last case was discovered. And we're going to see that in lab on Tuesday. We'll watch that story. Naegleria fowleri is a free-living amoeba. It's opportunistic, meaning if you go into this fresh, stagnant marine waters that have kind of been sitting in the heat for a while with no chlorine, and you jump in and you take a big dose of water up your nose, which everybody's had that, right? Everybody knows what that feels like. So that's the scary part, okay? So if that water gets into the nasal cavity and it contains the Naegleria fowleri, this amoeba can invade. And if it invades, it becomes primary amoebic meningoencephalitis, known as PAM, and this is bad, okay? This is usually death for the infected people. I think there's a handful now of surviving, and it's usually based on early recognition, having a drug that's experimental at the time. This little girl survived in Arkansas, but it was just all came together kind of thing. And I'm going to show you that story later. But swim and dive in those. So after watching these stories, I hope you will never jump into a freshwater source of water without closing your nasal, so you don't get that rush of water up your nose like you do in the swimming pool. Pathogenesis, invasion of the nasal mucosa, the cribriform plate and sinuses. This troph migrates through the olfactory bulb nerve and gets to the brain. And once it gets to the brain, it goes crazy. It starts giving headaches, fever, probably going into a coma state soon after. It is fast, it is aggressive, and it is deadly. It takes 3 to 6 days. Okay, so you may be jumping into a pool of water on Saturday, and by next Saturday, you're dead. Spinal fluid, spinal tap is the easiest, quickest way to see it. Brain tissue is usually after the patient has died. Okay, they can discover it then. So free-living amoeba infectious, you see that it has cysts in a freshwater sample. It has 2 forms. It can be an amoeba form, okay? And then it can be a flagellate form. It can have flagella. So it has basically two forms here. Okay, so it's just, gets up into the nasal cavity, invades the brain. Trophozoites will be seen in cerebral spinal fluid, and tissue flagellate forms in the cerebral spinal fluid. So we can see both forms in the patient. So diagnosis, we'd want to use spinal fluid, high white blood cell count in the spinal fluid, some RBCs in the spinal fluid, and we would all hopefully be able to see the trophozoites in the spinal fluid. Brain tissue, again, this is after death. We're not going to be staining brain tissue with H&E on a live patient. Culture, we can culture these, and this is unique for everybody who was in micro last semester. Put it on a non-nutritive agar, and then load that agar with E. coli. Okay, the trophozoite eats the E. coli and then cultures. It grows, it divides, it continues to do what it does on your plate, so you can actually culture this amoeba. So I thought that was really cool to bring in a micro, thinking about feeding it E. coli. All right, so the Naegleria fowleri, the nucleus, only one, no peripheral chromatin. The karyosome is large, irregular shape, and extends on most of the nuclear membrane. Karyolymph space is very small, so there's not this little pinpoint karyosome. The cytoplasm has numerous vacuoles. Inclusion, sometimes red blood cells, 10 to 20. Explosive formation, blunt pseudopodia, Addition of sterile water induces development and elongation of the two flagella. So that's the two forms. It can be an amoeba form without the flagella, and then you can actually induce it to form two flagella. So this would be the H&E stain, probably of brain tissue, seeing a cyst form. Notice the larger karyosome here. Some fluorescent look to it. So Naegleria fowleri cysts are never found in the tissue. Nucleus, only one. Same as the troph. Okay, so what is all of this here? This is tissue. What do we see? Troph or cyst? Troph or cyst in tissue? Yes. Okay, again, cysts are never found in tissue. Found in cerebral spinal. Nucleus, only one. Smooth cyst wall, average size, about the size of a red blood cell. All right, and I think we're running out of time. We're down to acanthamoeba, right? Is that our last one? So we'll hit this one real quick. I just want to show you the difference in the shape of the acanthamoeba. Yeah, okay. So free-living amoeba, opportunistic pathogen again. So we're going to see that this one's known as granulomatous amoebic encephalitis. And what was PAM for Naegleria? What was PAM stand for? Primary amoeba. What was the M again? Meningoencephalitis, yes. So acanthamoeba, progressive corneal ulceration leading to blindness. Okay. You know, did we see a little bit of that yesterday, right, on the video? Incubation period, 10 days. The disease is chronic, prolonged. Transmission can be inhalation of the cyst or traumatic injury with contaminated soil. Predisposing factors, it's usually this is an immunocompromised patient. Naegleria can be a healthy 12-year-old jumping in a pond. Acanthamoeba is going to be an immunocompromised host. Similar to Naegleria, but there is no flagellate stage for acanthamoeba. So the trophozoite of acanthamoeba compared to Naegleria is that it doesn't go into the flagellate stage. No peripheral chromatin. The karyosome is very large and darkly stained. That sounds familiar to Naegleria. Numerous vacuoles, but you won't miss acanthopodia, okay? Because I'm about to show you the spine life. So this is what acanthamoeba trophozoite looks like, and these are those spines that it's referring to. Again, a large nucleus, a large karyosome, little chromatin on the outside of the nucleus, large vacuoles, these spines and spikes for acanthamoeba. The cyst, trophs and cysts may be found in tissues. That's different than Naegleria, because Naegleria, the cysts are not found in tissue, only the trophs. So these would be the cyst, right? All right. We will try to look, I don't know what, I think we have Entamoeba histolytica for sure. We'll look at pseudoparasites on Tuesday, and then histolytica and Dipar. We might have both. I don't think we got Naegleria or acanthamoeba, but I'll double check. And if we do have any or all of those, we'll definitely pull those out to look at on Tuesday's lab. All right, I'm gonna stop the share. Gonna end the lecture. And y'all have a great weekend.